In order to be able to keep track of time, I propose that we can start. Our first presenter is Mrs. Valérie Gagnier, and the title of her presentation is "The Proteolytic Profiles of Lactic Acid Bacteria Drive Positive Interactions in Co-Culture in a Food Mimicking Medium."
So good afternoon, everybody, and I thank the organizing committee to give me the opportunity to present this work that is part of the PhD of Fanny Cannon. So as you know, the lactic acid bacteria can add up various functionality to develop new fermented food. Actually, we can preserve the food fermented products by production of various organic acid. Okay, do you see my screen?
Yes, yes.
So sorry for that. So I restart. So thank you for welcoming me in this symposium, in this congress, and I will show you the main part of the project, of the PhD project of Fanny Cannon. So, as you know, the lactic acid bacteria can add up various functionalities for developing new fermented food, and they can preserve the fermented food with the production of various organic acid, with the production of bacteriocin or antifungal components. They can also enhance the sensorial profile through texture and through the production of various aroma compounds, and they can change the nutritional profile and provide health benefit through the carbohydrate and protein hydrolysis, through the vitamin production, and through also the reduction in nutritional compounds. But the superstrain doesn't exist, so we have to associate lactic acid bacteria in order to have these various functionalities. So we chose a strategy in order to increase the positive interaction between lactic acid bacteria, and we propose to use nitrogen metabolism for doing that. So the objective was to enhance the interaction between lactic acid bacteria in a food mimicking medium and on nitrogen-based dependencies. So we chose to use proteolytic strains that are able to provide peptides and free amino acid in order to enhance the growth of proteolytic strains. So we wanted to know if it was the case, and if they were able to enhance the growth, if there was any specific peptides or amino acids that were implied in this. And finally, we want to know if in culture we can have enhanced outputs from the two partners, and if there were any change in terms of bacterial counts, acidification rates, amount in free amino acids and peptides, residual sugars, and volatile compounds. So the strategy was to develop a chemically defined medium that contained all components needed to ensure the growth of the bacteria, and notably we use only proteins as the sole nitrogen source. So notably we ensure the growth of only the proteolytic strains. Here is the presentation of the graph of the growth of the different proteolytic and non-proteolytic strains. So the proteolytic strains are growing as expected at a high level of population, while the non-proteolytic strains can hardly grow in this medium, and they only grew when we supplemented this CDM with tryptophan, that means with peptides and free amino acids. So we also used compartment chambers to identify positive interactions between proteolytic and non-proteolytic strains. So the different strains were separated in the chambers by membrane filters. So it facilitates selective bacterial count during culture. It gives information whether the contact between bacteria is needed for the interaction, and also it facilitates peptidomics investigation through metabolite exchange between the chambers. So here we can have the information on the growth of the selected strains in co-culture and CDM. So here is the example for one, the non-proteolytic lactococcus strain, sorry, and alone it doesn't grow as expected. The lactococci reach a very high population with the F proteolytic strains. It was lower in the presence with the L1 strain, and there was no growth in the presence of the L2 strains. There was the same tendency for the two other non-proteolytic strains, P and P2. And we can see here that the type of interaction depends greatly on the type of the proteolytic strains. We saw a very high interaction between F, medium interaction with L1, and no interaction with L2. And we checked for this strain that it was not due to a bacteriocin production in the CDM. So, as you can see here, there was a very distinct proteolytic profile for the different proteolytic strains in terms of peptides in gray or in terms of free amino acids in green. And we can see that there are very different profiles, and the type of interaction cannot be explained either by the quantity of peptides produced or the free amino acids. Actually, when the content in peptides is very high in F, in contrast to the other, and in terms of free amino acids, it was higher in L1. So we tried to check if it was from specific peptides or free amino acids. So in order to determine the peptide produced, we used the peptidomics. And here is the peptides that were identified. So there were more than 1,900 different peptides that were identified, and they contain from 6 to 50 amino acid residues. Almost half of the peptide produced were by the F proteolytic strains, and we can see that less than 10% were common peptides. So we here are represented the different peptides that we made some cluster in order to show their characteristics. So they can be grouped into 5 clusters, and we have the C1 and the C3 cluster that were peptides that were common for the 3 proteolytic strains. And after we can see with the C2 and C4, they were specific from the F strains, and the C5 for the L2. There was no specific cluster regarding L1. Here we also determined the main characteristics of the peptide produced by the proteolytic strains and those that were used by the non-proteolytic ones. First, peptides implied in the interaction were in clusters specific to F, and they contain preferably amino acid residues that were more polar, more non-polar. They have small side chain, and they have branch chain and aliphatic amino acid residues. Regarding, secondly, the peptides that were considering used by the non-proteolytic strains, they were considered as used if their abundance in co-culture were lower than in monoculture of the proteolytic strains. As an example here for the lactococcus strain, non-proteolytic, when she was co-culture with the F or the L1, we determined that only few peptides were specifically used. There were 62 in the presence of F and 21 in presence of L1. And the peptides were used preferably when we high molecular mass and when we have when we contain mainly branch chain amino acids. We also have difference in the different types of peptide that can be used between the different non-proteolytic strains, that is P, Plantarum and Lactococcus. And we also determine the content in branch chain free amino acids. And we thought that there are a large amount in these types of amino acid produced by F that was largely higher than for L1, and that was largely higher than L2. So we can conclude that the interactions that were possible with L1 can be possible through the production of free branch chain amino acids in contrast to L2. So finally, we co-cultured the different, we have the different characteristic at the end of the fermentation in the CDM. And here are represented the overall outputs that we show. And here are presented also the different distribution of the culture according to the carbohydrate consumed, the acidification parameters, the proteolysis in terms of peptides and free amino acids produced or consumed, and the 12 volatile compounds that were identified. So what we can see is that clearly the co-culture of the non-proteolytic and proteolytic and the proteolytic strains L2 was only the outputs corresponding only to L2. That confirmed that there was no interaction at all. After, when in the presence in the co-culture with L1, we can see that we have small outputs that were different in co-culture versus the monoculture. And finally, we found here with the F monoculture compared to the F co-culture with the different non-proteolytic strains that there was a great enhancement of the outputs produced, and there was a various different characteristics obtained at the end. So, as a conclusion, we can say that the interactions are driven by the proteolytic activity of the proteolytic strains, that it is essential to have the production of both peptides and amino acids. And the composition in branch chain amino acid is crucial, and the bacteria needs the isoleucine, leucine, and valine either in free forms or inside the peptides. The proteolytic strains then do not use the same types of peptides and free amino acids. And as a main point, the stronger the intake and the interactions, the more outputs are observed in co-culture of proteolytic and non-proteolytic. So I thank you all my colleagues for this work, and I thank you also for your attention.
Thank you very much for your very interesting presentation. We have time for questions, if there are any. I don't think that there is any question by the audience.
I don't see any.
So thank you again.
Thank you.
Have a nice meeting.
Thank you.
Okay. Our next speaker is Mr. Alexander Gmeiner from the Technical University of Denmark. And his talk is predicting virulence of Listeria monocytogenes using whole genome sequencing and machine learning.
Okay, that works. I hope you hear me well. Again, my name is Alexander Gmeiner. I'm a second-year PhD student at the Technical University of Denmark. And I'm a bioinformatician, so I'm actually one of these people who like to sit in front of the computer and do some cranking some numbers. I'm very glad also to be able to share some of the research I've been doing the last 2 years with you, which is mainly about predicting the virulence of Listeria monocytogenes using whole genome sequencing and machine learning. So why is Listeria concerning? I'm pretty sure many of you know how Listeria monocytogenes is a great threat to the public health, mainly because its high fatality rate of people infected of around 20 to 30%. It is also, how we heard in sessions beforehand, very resilient to different environmental surroundings, meaning it can withstand different pH levels and also salt concentrations. And one of the most concerning issues, I would say, is that it grows well at refrigerated temperatures, which makes it very concerning for food products in general. So what can we do against Listeria monocytogenes? We can do pasteurization, which is a great tool against a variety of different foodborne pathogens, and it works well for Listeria. However, there's a little problem because there's always the possibility of the recontamination along the production line. And for example, for some food products, as raw milk cheeses, for example, pasteurization is just not part of the production process. So hence, a lot of countries have actually implemented 0 to very low tolerance policies, which actually look at Listeria monocytogenes on a species level. However, current research finds more and more evidence, and for example here by Mori et al., that the virulence on a subspecies level is quite heterogeneous. And also you can make association of different subtypes of Listeria monocytogenes to specific niches, meaning they are found very often in food, but maybe then not in the clinical setting. Further, whole genome sequence surveillance is also gaining more and more popularity. It's already like the common standard in the clinical setting and in means of One Health. And thinking about One Health, surveillance is more broad and includes different sectors such as the food industry as well. So what are we actually doing in our project? We are using these whole genome sequences from governmental surveillance systems in combination with machine learning to predict and try to predict the virulence of Listeria monocytogenes on a subspecies level. Here you can see a little overview, and I don't know if you can actually see the pointer, but you can, yes, great. So here's a little schematic overview of the entire project. So we gather our sequences, we gather some feature from the sequences, put it into a format that can be used by machine learning to actually analyze the data, and then we can make predictions about samples that are new that we find. So the data that we're using for our study comes from 2 countries, from Denmark and from France. Denmark and France have a very, very tight surveillance systems of Listeria, and it's very controlled, meaning that all the samples that are found in a food context and the clinical context are whole genome sequenced and then put in a database that can be accessed with special permission. What we also need to think of is how we actually quantify the virulence. There's many different ways, but we're using a little bit a more qualitative approach. So what we're doing is we gather our samples, we classify them using multi-local sequence typing, and then we gather a measure which is called the clinical frequency from this data. And the clinical frequency basically just describes the number of the clinical isolates over the total number of isolates that are found, meaning in a clinical setting and also in a food setting. And the higher the clinical frequency, the more virulent, in a way, are these Listeria in our hypotheses, because there needs to be, or we hypothesize that they have an advantage if they make it to the clinical setting. After that, we are actually exploring 6 different machine learning models. They're also called algorithms. I will use these 2 words interchangeably from now on. And I will not go into great detail with machine learning because I'm pretty sure you don't know a lot about it, and it gets very detailed very quickly, which we don't need today. But what I want to focus on is actually a little bit of a big overview of how it's used. So we cannot actually input the whole genome sequencing data into machine learning because our models will not be able to gather a lot of information from such a huge amount of sequences, or not the sequences per se, but just from that data. So what we're trying to do is we gather some features from the whole genome sequences that we think are good in predicting or help to predict the clinical frequency. So these features can be different things. It can be, for example, absence and presence of genes. It can be pangenome genes. It can be absence and presence of SNPs. And then for each of the samples, we put them in our feature space. And then what we also do is we know our clinical frequency. And this we all put together into a matrix and give it to the machine learning algorithms. And they will then train on this data, and we will select a best performing model. And then when we have this model and we get new data, we can translate our new data into our feature space, for which we do not know the clinical frequency. We give it to our model, and our model will be able to make predictions about the clinical frequency. So if we look at some results that we have had, here you see a performance plot. When I talk about performance, it basically just describes how well is our model in predicting the clinical frequency. We have here comparisons of different genomic levels. We have a set of virulence genes, a set of pangenome genes, and also a simplified version of SNPs analysis. And what I want you to focus on here is that when you look at the performances of these different models, you can see that they all perform similarly well. So there's not a model that sticks out that has like a great performance. But what we can also see is that if we use the pangenome genes, there's actually a higher performance. On the other hand, if we use the SNP genes, there's a lower performance. This is great, great results. It seems to be working. However, we have to take these results a little bit with a grain of salt, because if you remember beforehand, we only have 169 samples, which for machine learning is not a lot at all. The power of machine learning is really in like big data and a lot of samples. So we were trying to look for more data. It's not very hard, not very easy to get surveillance data from different countries. There's always very tricky. So we thought maybe we can trade a little bit of the accuracy we think that is in the governmental data because we're using the data itself to create our clinical frequency measure for more data. So we're looking into public data available, and we have found the Genome Tracker Network that combines a lot of governmental institutions in the US and also some collaborators. As said, the data is mainly focused on the US, but there's other countries contributing as well. But we are only in this pilot study focusing on the data from the US. There is about 26,000 sequences on the platform, which is a lot of samples. If we do some filtering, we end up with 2,800 samples, which is still great and way more than our initial 169. If we look at some results that we get from the Genome Tracker Network, we still have our performance plots over the different 6 algorithms. I have to compare, for comparison, I actually put also the data or the performance measures from the governmental data, which will be the darker shaded colors, and the lighter shaded colors will be the Genome Tracker data. And what you can see here actually is that the landscape of the Genome Tracker data for the performance is more diverse than it was for the government, meaning we have some models, for example, the random forest model here. Actually here, you can see that there is an increased performance. And what you can also see is actually that there's not as big of a difference anymore between using virulence genes as features and also pangenome genes. So as a little take-home message here from me, I want to point out that whole genome sequencing in combination with machine learning actually seems very promising to predict the clinical frequency of Listeria on a subspecies level. We could also see, which also has been mentioned in our introduction lecture, that more data can improve the predictive performance of the machine learning. And here I put good data because it's not necessarily more data, but like more diverse data helps in definitely in predicting performance. And also, One Health whole genome sequencing surveillance data is a very powerful tool. So there's a lot of information stored in this data, and it's very great that we're moving towards more of a One Health approach in surveillance as well. And at the end, I would like to thank the scientific committee for giving me this opportunity to talk today. My funding buddies, a special shout out to my supervisors, Patrick, Lisbeth, and Shinny, and also the entire group. Thank you.
Thank you, Dr. Gmeiner, for your nice and quick presentation.
Thank you.
Are there any questions from the audience? No? And I see there is one here. Yes.
I don't know. I can speak loud.
Yeah.
What it triggers me is the fact that you're actually testing different algorithms to understand which one is working best.
Yes.
You don't think that you are actually introducing a bias at the moment you're choosing one, in a sense that if the approach is there, I mean, again, I'm totally ignorant on the basis of the algorithm, so as you said, well, I'm not entering in here because we were not able to get out of it.
Yeah.
But if the prediction, or let's say that if we want to mimic, or if we want to reach a kind of prediction, you don't think that any algorithm should get to the same point of giving like a prediction if it is possible to get there? I mean, I'm always, you know, let's say, a little bit puzzled with also biostatistics, that at the end of the day, you are going towards an approach that gives you the actual result that you want to get. So how is this kind of bias, or let's say, I will not say bias, but guidance, or entering into the prediction?
Thank you for this question. It's a very good question, actually. It's definitely tricky. But I think in machine learning, all of these different models have like different strengths and weaknesses, let's say, as similar as you use different methodologies if you want to shine a different light on specific things in the lab, right? You use the methodologies that you think are appropriate to give you the results. It's the same here. It's kind of like finding the right parameters, and all these machine learning models have different parameters. If you tweak them right, they give you a good outcome. And also because it's not just the parameters and stuff, but it also depends on the data itself. So how is the data related to each other? How are the different features related to each other? So I don't think it's like just cherry picking what is working best. It is kinda, but it also is that for different types of data, different algorithms work better, and they're designed to work better for different sets of data. So you're just choosing basically what's appropriate for the data and not just what you want or what gives you what you think it is. Yeah.
Yes.
Maybe I misunderstood something, but when you evaluate the performance
Yes.
of the different algorithms, what is the reference you are using? I didn't understand exactly what you are using as a reference
Yes.
for the variance.
Yeah, if I understand you correctly, how we get like the comparison if the performance is good, right? Is that what you're asking? Yeah. So we are in a sector of machine learning that is called supervised machine learning, meaning if we like give data to the models and we train the data on, then we know the outcome of the data. So we know our clinical frequency for each of the samples, and then we train our data. So we give the features to our model. The model makes a prediction, and then we compare with the real outcome.
So only the frequency provides the variance.
Yes, in our study, we're using the clinical frequency as a description of virulence. Exactly.
So it's per sequence type.
Exactly. Yes, it is on a sequence type level. Yes. Yeah. I should have made it a little bit clearer.
Another
Yes,
one?
another.
I have a question regarding your observation that looking at the band genome, your model is better working than the
Yes.
virulence genes, which is somehow surprising. So can one estimate from your results that in the band genome there are more virulence factors hiding?
I mean, that would be great. I don't think this is the case here because what I think is happening, it's more like a technical thing, right? So how I choose, I cannot choose the entire pangenomes because I will introduce bias if I use thousands and thousands of features with only 100 samples. That's not possible. So I need to filter them beforehand. And I filter them, I look at all of the percent identities. Actually, I do a blast search, and then I look for the pangenome genes that have the biggest variety within them, right? So that doesn't necessarily mean that they are good predictors of like virulence or like virulence factors. It's just like saying, since there's a big variability between the samples, it's just easier to get like a pattern for the different models when they're learning. I hope that answers your question.
Well, if there is any other question, I have one, if I may. You said that the reason that you use the virulence frequency parameter as an index of virulence potential was based on the assumption that strains that or isolates that made it until the clinical level were most likely more virulent. So I was wondering in the number of food isolates parameters, knowing that the infectious dose of Listeria monocytogenes, at least for healthy individuals, is expected to be high. If the concentration in the food samples is not taken into account and just we take into account the number of isolates, do you think that this may be introduced some kind of bias in this index you are using?
So I think maybe biased, yes and no, I would say. It's just a different way of looking at it. I feel like there's many different ways, right, why an isolate makes it to the clinical and is like actually infecting a person. There's like a lot of factors that are like maybe beneficial to like endurance in like the food industry, and then also there's many factors between it, right? So I don't think we're introducing a bias per se. We're just like using an overview and like basically zooming out, looking at what is happening, and then using the data that we have together and like to make try a figure or like a measure of how virulent or how dangerous they are actually.
Okay. Thank you.
Thank you too.
Thank you very much.
Thank you. Okay, next speaker is sitting next to me.
No,
No?
I don't think so. I think next speaker is Dr. Huriye Mohammedpour. And she's from University of Padova. And her presentation is entitled A Study on the Community of Archaea in Food Matrices, along with the investigation of living archaea in one of them.
Hello everyone, I am Huriye, coming from Italy. I'm doing my postdoc fellowship in the University of Padova, and here I am going to present a summary of my research that was done on prevalence of archaea community in several food matrices. First of all, we will have a brief overview on the history of archaea. Archaea are known as the third elementary domain in the tree of life, which is located between bacteria and eukarya, due to several common features with these two domains. The most special characteristic that make these taxa become different from the other microorganisms is being extremophile, which means they are able to grow and live in extreme environment, especially in marine environment, such as environment with high salinity, high acidity, pressure, and temperate environment, and anoxic environment that filled with methane or the other gases. So based on their habitat preference, they were categorized in three different groups of hyperthermophilic, extremely halophilic, and methanogens. But over the years, with progress in molecular approach, investigation in presence of archaea in the other ecosystems received more attention gradually, which resulted in discovering more taxa in the other ecosystems. And interestingly, data obtained by DNA sequencing of human microbiome revealed the presence of archaea in human gut and oral microbiome. And this was the first insight into making a hypothesis that archaea might be present in human diet. So the first studies with this aim initially started with traditionally salty and fermented foods because they were expected to harbor more archaea respect to the other foods. But despite of discovering extremophile archaea in these sectors, still their presence in the other food matrix become unexplored. So in this study, first of all, we aim to investigate the presence of archaea in several food matrix by culture-independent methods, and to have information on the community of archaea and its relationship with foods. As the second step, we attempt to evaluate how it is possible to culture archaea under normal conditions. To this aim, among the matrix, the one with a high prevalence and diversity of archaea was selected for further experiments. As the final step of this study, comparison of data obtained by culture-independent and dependent method enable us to, first of all, have an information on the community of archaea in several food matrix, distinguish the culturable and non-culturable taxa, and to get some information on the condition that's required for incubation for growth of archaea. All this data together with the other studies enable us to reach a comprehensive conclusion of the current study. Here, the experimental scheme of the first step is presented. A total of 7 matrix, including milk, cheese, honey, hamburger, trout, clam, and brine, were analyzed independently. After preparation of the homogenate, an amount of that was collected for DNA extraction that was done by DNA-Z power soil extraction kit. By using the specific primers that were assigned for archaea based on the published study, the amount of DNA was quantified by real-time PCR, and after preparation of the library, they were sent for sequencing and followed by analyzing by Kraken pipeline. The first results of sequencing revealed a significant high population of bacteria respect to archaea that we already expect such this result because, as previously mentioned, archaea have more preference in living under extreme environment rather than normal environment. But only considering the data of archaea among the samples, 3 groups of clam, brine, and milk were identified as the matrices with a high community of archaea insights. This figure shows the distribution of taxa at the genus level in each matrix. The blue color are corresponded to methanogens that compose a big portion in several matrices, except for brine that was ideally full of halophilic archaea. And the second rank was corresponded to unknown taxa of archaea. This fact also put more value in order to study, put more value on the importance of study archaea to discover more taxa in future research. The community structure of archaea was also characterized by richness and Shannon index. Based on the richness, the group of brine and clam were identified as the matrices with high community of archaea inside, and based on the diversity, also all the matrices showed much higher diversity respect to their richness, but also the group of clams showed a high heterogeneity among the other matrices. The amount of DNA that was quantified by real-time PCR was categorized in 3 different groups of negative, detectable, and quantifiable. That's in this study, we only consider the quantifiable as positive, and the amount is presented as log copy in figure B. But according to figure A, it could be understood that although the amount of DNA was not quantifiable in all the matrix, but at least all of them showed a detectable amount of DNA. Also, in the case of brine and trout that showed a high richness of archaea inside, all the amount of DNA was detectable and quantifiable. Considering this data together with the other data, with the previous data that obtained by NGS, revealed that among the samples, the group of clams showed a high richness and diversity of archaea inside. So it was targeted for the next step that was proposed to evaluate to evaluate the culturing of archaea under the normal conditions. This is the scheme of the experimental method. After preparation of the homogenate, an amount of that was collected for directly sequencing, assigned as CI, and the other amount was collected for classical culture method, including serial dilution, culturing, and incubation. The culture media that was used in this study was marine agar containing 3 antibiotics in order to minimum the growth of bacteria as much as possible. And for the incubation, we proposed 3 different temperatures of 4 degree, 22, and 37 degree, and 3 different duration of 3 days, 15, and 30 days to distinguish the sacrophile to mesophilic and the fast-growing to a slow-growing taxa of archaea. After incubation completed, the plates were washed with BBS and transferred to cryotubes for further molecular analysis that was done completely similar to the first step. The results of the sequencing are summarized in these 2 figures. Figure A corresponded to the homogeneous samples that they didn't follow the culture method, and figure B corresponded to cultured sample. The big differences between these 2 figures is that more taxa with different richness were obtained in homogeneous sample respect to less taxa with high richness in cultured samples. The main reason for these differences is that in this study, we provided the minimum requirements of growth condition for archaea. This is why that archaea are famous for specific requirements in growth condition, whether culture media with varieties of additive and supplements, and whether for incubation condition with a very long duration of maybe 2 or 3 months, and modified atmosphere. So, at least here we found that there are several types of archaea that are able to grow under minimum requirements in the growth conditions. A screening of incubation condition revealed that majority of taxa were grown at lower degree during a long-term incubation respect to 37 degree, that only a few taxa were grown in the initial days of incubation. Here we have a table with data that NGS in the first step of the experiment was not able to give us. Based on this data, that the taxa that obtained by culture method, they were categorized in different groups of only culturable, only detectable, majority of taxa that found culturable and detectable with preference in long-term duration, and several taxa that found only growth under a specific temperature. This table somehow shows the importance of having culture methods besides the molecular methods for having a complete or better results in future. For example, in our study, of course, we achieved new data on in relation to the archaea and their presence in the food matrix. by culture-independent methods such as the richness and diversity of archaea, common taxa present in each matrix, the matrices with more unknown taxa of archaea, and prevalent taxa in each matrix. But on the other hand, we reach new data by culture method, such as the community of living archaea, optimum growth incubation condition, fast-growing and slow-growing taxa, and culturable and non-culturable taxa. Even comparison of data with the previous studies, besides of showing similarities, also showed differences in some aspects. For example, despite of the other studies that claim archaea are present in extreme environment and they need a special culture media, higher temperature and long-term incubation for growth. In our study, we found taxa that could grow in very normal matrix with the ability in fast growing at lower temperature and in simple culture media. So at the end of this study, as a conclusion, here we highlight the importance of further investigation on archaea prevalence and anticipate that application of comprehensive combination of both culture method and DNA-based method will lead us to find out more about unexplored or underexplored archaea that exist in the environment. Investigation on new taxa or even prevalent taxa in environment lead us to find out their harmful and useful roles in different areas of industry, biotechnology, medicine, and human and animal and plant healthy in order to improve or prevent their growth. At the end of this presentation, I have to appreciate the best supervision of my supervisors, Professor Barbara Cardas and Professor Luca Fazzolato for their best supervision, and my colleagues, Dr. Lisa Carraro and Marco Cardi, for their best cooperations. Thank you for your attention.
Thank you for your presentation. We have time for a couple of questions, if there are any questions. Yes, please.
So my question actually are two. If you can give us indications on the load of archaea, so in terms of, let's say, thank you. So in terms of counts, for instance, because you have shown copies of 16S RNA, but for microbiologists like me, I want to see counts. So I would like to see, or at least a kind of let's say projection on, in your opinion, how many cells correspond to copies of 16S rRNA genes, because this is going to be important. The other thing is, if you have an idea about the roles, because you have put some things in the last slide, but it would be nice to have an idea on the roles. And actually, the other thing is that probably your study demonstrated that archaea are actually not only present in extreme environments, because when we are talking about milk, for instance, milk to me is not an extreme environment.
It's completely
so probably,
normal.
Probably this is maybe one of the outcomes that should be further investigated in order to understand a little bit better the ecology of this group that I agree with you is underestimated. Thank you.
Okay. First of all, your first question, you mean, I didn't understand very well that.
So in terms of counts.
You mean that the quantification of the DNA?
But what about in terms of,
detectable and quantifiable, you mean? Because...
so in terms of numbers of cells?
Yeah.
The counts that you have, the 16S quantification, the DNA quantification, does it correspond to, in your opinion, like to, let's say, 1 million per gram, 10 millions per gram, 1,000 cells per gram?
It was, the threshold was 1,000 or less. Because currently the big problem with Archaea investigation is that we don't have a completely specific primers to identify them. The primer that we and the other previous studies that used, this primer can at the same time detect both bacteria and Archaea. So we had this problem, but totally the amount of DNA was low. And we also categorize the amount that was detectable and quantifiable by the signal of real-time PCR. But in total, the amount was not too high. Maybe the maximum was near 2,000 reads. And your second question, the roles of Archaea. They have both harmful and useful till now. Harmful, like because several data shows that they have roles in pathogenesis. For example, in the guts, healthy, bad effect on gut, human gut healthy, some infection in mouth with mouth infection. And they have some good, useful effects in fermentation, improve the fermentation to producing flavor, and some bad effect on the spoilage of fermented foods. And also they are able to produce carotenoid and peptide with antibacterial effects. So because of this, considering these two effects, made us to bold this topic for further future, for further research in future.
Thank you.
It's okay.
Are there any questions? No. Thank you.
It's okay.
Thank you. And now is the time for you. Okay. So next speaker is, was sitting next to me is Dr. Alexandra Lianou from the University of Patras, Department of Biology, and she will present a work in collaboration with the Agricultural University of Athens, Fate of Salmonella enterica in tahini sesame-based products.
Well, good afternoon once again. My presentation will be on a food hazard combination that has attracted our attention during the last decade. Very well known, notorious, I would say, but still hard to deal with pathogens, Salmonella enterica, and a food product, sesame paste, commonly known as tahini, which has an increasing popularity among consumers the last years. Most likely, most of you in this room are familiar with the pathogen Salmonella enterica, so I will not take much time introducing this organism. I will just highlight that some of its basic features that make this organism a difficult to solve puzzle still is its numerous serotypes, its potentially low infectious dose, which may be fewer than 10 cells in certain food products, its robustness against a wide range of environmental stresses, and of course its extensive antibiotic and antimicrobial resistance. According to surveillance data reported in the 2020 zoonosis report of the European Union Health Zoonosis Report, salmonellosis was the second most frequent zoonosis next to Campylobacteriosis, being associated with 22.5% of outbreaks, almost 50% of hospitalizations, and 16.7% of deaths. With specific reference to foodborne illness outbreaks, these were reported to be 694, resulting in more than 3,500 cases of illness, 812 hospitalizations, and 7 deaths. Similar is the picture as reported by the United States Centers for Disease Control and Prevention, where Salmonella was the second after Campylobacter in terms of incidence, but first one in terms of hospitalizations and mortalities. Several food products have been related to salmonellosis, outbreaks and sporadic disease, including the very well established and traditionally associated ones like the eggs and the poultry products and the pork meat products, but also involving fresh produce commodities, confectionery such as ice cream, and of course low moisture foods such as peanut butter and other nut butter spreads and similar products, breakfast cereal, and chocolate products. Actually, it has been reported that the majority of reported outbreaks related to low water activity foods is due to the contamination by Salmonella. And a great example of this is the recent multi-state, multi-country outbreak of salmonellosis, which was associated with contamination by Salmonella typhimurium. It was a really rapidly evolving outbreak that affected 13 European countries. And children, especially under 10 years of age, were most at risk for severe infection. So this group of products, the low moisture food products, and particularly the ones with high fat content, have very scientific interest with regard to the survival of pathogens and Salmonella enterica in particular. So one of these products in this food product category is actually tahini, a sesame paste product, which is a ready-to-eat product that has been traditionally consumed as a condiment in Middle East and also the Mediterranean region, but it has gained popularity among consumers in other areas of the world, including the United States, Canada, and many European countries. This is due to its high nutritional value. It's a very nutritious product, rich in polyunsaturated fatty acids, vitamin E, lignans, and also protein. It's usually manufactured from dehulled, ground, and roasted sesame seeds. It has a long shelf life due to its very low moisture content at ambient temperature, which can reach up to 2 years. And it can be consumed either as is or as one of the ingredients in the preparation of other ready-to-eat foods, which are also increasingly popular, such as hummus and various salads dressings. Several foodborne outbreaks during the last decade and recalls have been related to tahini and tahini-based products. And the origin of Salmonella enterica in this type of product can be either its basic raw ingredient, in other words, sesame seeds, which are susceptible to microbial contamination during their growth, storage, and processing, of course, providing that the contaminants are able to survive the applied roasting process, and can be also due to post-process contamination during production and packaging due to poor hygienic conditions. These are some typical basic steps in the manufacture of tahini, where in addition to roasting, critical control points are also regarded in this process flowchart, the grinding step and also the packaging step for the reasons that I just mentioned, because they may act as ports of entry for the pathogen in this type of product. This figure here, which has been taken from a recently published article, what actually shows is the relationship between the different serotypes, the countries, the involved countries and food products, and of course the year of occurrence of salmonellosis outbreak related to low water activity foods. And what we can see here is an increasing trend in these outbreaks over time. The involvement of multiple countries and serotypes, and of course a strong association of sesame paste-based products with these kind of outbreaks. So the objective of the study that we conducted was the evaluation of the survival potential of Salmonella enterica in different tahini products during storage at 2 different temperatures, refrigeration and ambient temperature. For this purpose, we used 3 different products, one regular tahini product consisting exclusively of ground sesame, a second product that was sweetened by honey, using honey at concentration of 20%, while the additional ingredients are shown here of this product. And the third product was a product that also contained fructooligosaccharides at a concentration of 10%, and was sweetened with stevia glycosides. The information provided in the nutrition labeling of these products is summed up in this table. These 3 products were inoculated with a 4-strain composite of Salmonella enterica, and along with non-inoculated samples were stored at 4 and 25 degrees Celsius for a total of 90 days. At regular time intervals, microbiological analyses were performed using duplicate samples corresponding to distinct product jars, different jars, and microbial enumeration was performed on both XLD agar, a selective medium, and Trypton soy agar, a general medium, in order to determine the healthy and the total survival populations. Also, pH measurements and water activity measurements were conducted. And we move to the results. Here we see, I don't think that I can use this. Here we see the reduction, the log reduction of Salmonella enterica populations in the 3 different products that we tested during storage at 4 and 25 degrees Celsius. One observation that was made was that a better survival of the pathogen was noted during storage at refrigeration, which is something that has been also reported by other researchers in similar products. Another observation was that in both temperatures, the highest inactivation was observed in the regular product, of course, and the highest survival in the product with sweetened with stevia, which is here, and particularly this was evident during storage at 25 degrees Celsius. And always annoying observation to make is having big error bars in your graphs, which we had it here in the regular product, which as annoying as it is, it always indicates something. And to my point of view, in this case, indicates most likely uncertainty that was inevitably introduced in the measurement because of the high viscosity of this product. Because this product was pure sesame seed grounded, which means that we had phase separation during storage, and it was more difficult also to inoculate and also to be sure that you have a homogeneous population dispersion of the inoculum and also during sampling. So despite this, however, we had some good indication about the highest inactivation of the pathogen in this product. And what we also saw in the previous graph, and we can also observe here, when we can see the percent survival in the 3 different products and at the 2 different temperatures, there was a higher survival in the product with stevia that was particularly evident during storage at 25 degrees Celsius. This finding was also in agreement. Something like that was not observed, such a distinctive behavior of the pathogen during storage at 4 degrees Celsius, where a similar survival was recorded for the product with honey and with stevia. And these observations were actually in agreement with the observations made looking at the graph showing the overall percent injury of Salmonella enterica in the different products, where although at 4 degrees Celsius, similar level, at least they do not seem to be statistically significant, levels of injury were noted. During storage at 25 degrees Celsius, this was drastically changed, and we saw a clear distinction of the 3 products, with the highest level of injury being observed in the product with honey, the lowest in the product with stevia, and somewhere in the middle was the regular product. In these figures, we can see the changes of pH in the different products and the 2 different temperatures. The continuous lines correspond to the inoculated products, and the dashed lines to the non-inoculated ones. With regard to the non-inoculated ones, I forgot to mention that actually Salmonella was not recovered at detectable levels in any of the products. And by looking at the graphs, we actually see that no considerable changes occurred during storage of the products, and neither the inoculation nor the storage temperature appeared to have an effect on what was measured in terms of pH. In this table, we can see the water activity values of the three different products. The initial ones in the middle of storage and at the end of the applied storage. The lower water activity values corresponded to the regular product, which somehow explains the highest inactivation that was recorded in this product. However, we could not fully explain the behavior of the pathogen based on the water activity values for the other two products, where the product that actually supported the survival of Salmonella enterica, the one with stevia, had lower water activity values than the one with honey. Another observation made was the gradual decrease in all products of the water activity during storage, but the applied storage temperature did not appear to have an effect, a considerable effect on this. So in conclusion, the survival of Salmonella enterica in tahini appeared to be affected both by the product type and the storage temperature, with the highest inactivation at both temperature being recorded in regular tahini, and a better survival potential being noted overall during storage at 4 degrees Celsius than at 25 degrees Celsius. Finally, the higher survival and a lower level of injury, particularly at 25 degrees Celsius, was recorded for the product that was treated with stevia. The observations made could not in full be explained by the pH and the water activity of the product, which means that these observed differences are most likely associated with the food matrix composition and structure. These three products were different products, and actually the one with the honey had like 3 or 4 more ingredients compared to the other one. So it's not only the sweetening agent that differed in these three cases. And by all means, additional experimental data are needed in order for sound conclusions to be drawn, and this should originate from independent experiments with distinct product batches, and then we can proceed to the accurate determination of inactivation kinetic parameters, and such type of information, along with research that specifically aims at the elucidation of the role of the product's constituents in their microbiological safety, should certainly be very useful in microbial risk assessment in these types of products. As fascinating as it may be to look at the real food product, because what you see there is what actually is expected to be seen in real life, when you observe something there, then it's not easy to interpret it. So at this point, only assumptions can be made with regard to why the product with stevia actually supported a better survival of the pathogen. But actually, this is a good information for someone to look at its constituents and do some model food systems maybe to elucidate this effect. Additional research areas that are certainly very interesting to be tackled include the investigation of the mechanisms that maybe underlie the survival behavior of Salmonella enterica in low water activity food matrices, and of course the characterization of the contribution of viable but non-cultivar state in this behavior, which was not taken into account in this experiment. With this, I would like to thank you for your attention, and if you have any questions, I will be happy to answer them. Thank you.
Question, please. Yes, at the back.
Yeah, so you said that you thought that the difference in Salmonella was due to the viscosity of the products, but could it just be that the extra addition of sugars made it easier for the bacteria to grow?
I cannot hear you well. If you could please repeat your question. I heard the first part.
Could the reason that there's more Salmonella in the products with sugar be that they have access to easy nutrients and therefore grow faster?
Well, for the first part of your question, I just said that maybe the uncertainty involved is higher in the regular product because of its viscosity. It may play a role also in the survival, but I don't think that this was the case here. The case was that it was a very, very low water, the water activity was very, very low in this product in terms of surviving. With regard to the other products, under these conditions, under these low water activities, we do not expect very active metabolism by the bacteria. So it's not a matter of, growing or utilizing a lot of the available nutrients in their environment. Beyond that, I could discuss something like this for the product with honey, which actually has sugars that can be used. But in the case of the product with stevia, neither stevia nor the dietary fiber that was incorporated in the product can be actually utilized by salmonella. So I don't think that this might be the reason here in these conditions.
Thank you. Thank you.
You're welcome.
And maybe I am not sure to have well understood, but seems that salmonella survive better at 4 degrees than 25.
Mm-hmm.
If I look at the standard error in your graph, if
I don't know if I can.
we have a look of standard error, I don't understand.
Okay,
How to
let's
conclude about better survival at 4? So can you tell me more, please? And
Yes.
second question is about strains you used.
Mm-hmm.
This better survival can maybe depend by the strains, the type, the kind of strains you used.
Okay, I will start from the last part of you. Everything depends on strains. And that's one of the most important decisions that maybe one has to make when designing an experiment, because selecting the strains is always... so there are certain recommendations regarding this, and what is usually done when we don't have enough information or when we don't have, let's say, available strains originating from this specific food matrix is to actually use a cocktail of what we have. So ideally, the strains that we use, which we were for in order to meet these recommendations, since we don't have enough information, these strains ideally should have been, let's say, tested for their maybe, I don't know, desiccation survival, osmotic resistance, something like that. That was not done in this case. They were just used as a cocktail in order to avoid, any bias that would be created by using a single strain. So regarding the first part of your question, what we actually do here is to compare the. It's not what I said. It's the trend that we see. And actually focusing on the first 20 or 30 days of storage, where even for the black line that corresponds to the regular product that had the biggest variation, we can see a very high slope during storage at 25 degrees Celsius. I haven't performed because this experiment was performed only once. I didn't move to the statistical, let's say, analysis, but in terms of inactivation kinetics, I think it's obvious, at least for the one that is actually getting activated, the black lines, that there is a clear effect of temperature. And the same applies for the other two. I'm sure that if we proceed with, let's say, inactivation rate estimation, there will be some really considerable differences there. And the higher the inactivation, the higher the difference. The resistant ones maybe not so something, considerable. The explanation to this has been observed. It's not the first time that we observe this. Is that the low metabolic rate that low temperature results to in, let's say, non-psychotrophic organisms. Salmonella is not a psychotrophic. Actually protects them against additional stress And that was not surprising as a finding, but it was good that we actually confirmed previous findings regarding this. And it's actually very interesting because consumers may get into this selection of temperature, feeling that it will protect them more. But in this particular case, it's not a good idea, maybe.
Thank you. Yes, Luca.
Just on one point on the strain selection, you have used 2 strains from animal origin. How would you think going into strains from plant origin would affect would have an effect, or who cares at some point?
I would guess that plant origin strains would have a higher possibility of being more resistant to adverse conditions. So maybe I'm not sure, we would get some more robust survival behavior. Of course, it always has to do with the strain interactions occurring in this mixture, which we don't know how they will interact. But yes, that would be a better selection of strains.
No more questions. Thank you, Dr. Olianou. And please, Mrs. Dimitra Tsoureki, PhD student at the University of Torino, will present her work on exploring associations between metataxonomic profiles and pathogen presence along infant food production chain.
Thank you. Hello, everybody. Thank you for being here in my presentation. So let's start. This chaotic slide is on purpose like this because I want to point out the fact that if we focus on the findings of the European monitoring system of detection of foodborne pathogens in specific, in a monthly base, that is the month that we are on, we can find focusing in the 2 main foodborne pathogens, Salmonella and Listeria monocytogenes, Salmonella species and Listeria monocytogenes. We can find them almost in daily basis in different European countries and from origin that is coming from around the world. So we can understand that this is a really big burden, as World Health Organization is showing with this infograph on your left, excuse me. And this is also a burden for our group of interest that are the children with less than 5 years of age, that they are facing this impact because 30% of their yearly death is because of foodborne illness, foodborne disease. So the purpose of this study is to figure out how the pathogens enter and persist inside the infant food chain, understand the composition association in the food microbiome of this chain, and try to figure out correlation between targeted and untargeted methods. Everything starts from classical microbiology, that practically we, for both foodstuff and environmental samples, we focused in an enrichment approach where we wanted to detect 5 pathogens, foodborne pathogens, 4 aerobic, Listeria monocytogenes, Bacillus cereus, Salmonella species, and Staphylococcus aureus, and the anaerobic Clostridium perfringens. For that reason, we placed 2 time periods. The time 0, as you can see, is the moment that we dilute our food or swab, in the case of environmental, in a generic broth, in the case of the aerobic, and in a specific selective one for the Clostridium. And there we are keeping aliquots in order to further analyze them by molecular and chemical analysis. In the time 24 hours, we are getting also aliquots, but also we are performing isolation in selective media for each of the pathogen. Further, passing to the molecular techniques that we have used in this study, after the DNA extraction, so we are focusing on DNA, we are using untargeted methods, meaning metataxonomic analysis with the use of Illumina MySeq technology, and a targeted method with a real-time PCR using TACMA probe technology. Here you can see the flow chart of the company, and in few words, we have divided as an incoming area and as a production area, upper and down part of the flow chart, where in the incoming area we were collecting the raw material and the production, all the other, intermediate final products and environmental in different areas of the production. In total, 193 samples were collected. So we started to find a differentiation between the microbial composition. If we stayed just in the type of sample, so environmental and the 3 types of foodstuff, but also seeing them collectively, we cannot see any variability in between the samples. If we look on the type of raw material, we can see that a little differentiation seems to appear in the milk powder, for example, that are the blue dots, and then all the other raw material. Passing to the other 2 foodstuff, so intermediate as IP and final product as FP, we can see that we have a differentiation, a little differentiation on time. As you can see, campaign 1, excuse me, I forgot to say that. We have divided our experiment in 3 campaigns. The campaign 1 is it's from October to December of 2021. The campaign 2 is from January to April of 2022. And the last campaign, it was the summer of 2022. So what we can see here is that we have a little differentiation in the food products, but this is not secure and it's not also significant to say because we have few samples. But if we see, with the same idea of campaign division, the raw material that are higher in amount, we can clearly see the differentiation on time and on origin, so type of raw material, in between the 3 campaigns. Even more clear if we see the environmental samples on time, a long time, that you can see from the left to the right, so from the blue dots to the yellow, how the microbial diversity is changing, so is forming little groups. So taking in consideration all these factors, we have visualized our metataxonomic composition with the red lines, dotted lines. You can see the different time points in between these different campaigns. If we stay at the lower level, the phylum level, and in class level, practically in between the red lines, we can see a kind of oscillation. So there is the same amount, more or less, of firmicutes as the most present, most abundant phylum, and then is coming the proteobacteria. The same idea, the same way of viewing is also for the class, that the bacilli class is the most abundant, followed from the gamma proteobacteria, and fourth are the Clostridia class that contain one of our pathogens of interest. The families became already more complicated, but also here Bacillacea remaining the most abundant, and we can still see Staphylococcaceae, Clostridiaceae. But going more into deep, so at genus species level, it's always following the same idea of time, a long time, so we are seeing the profile in a long time. You can see that in campaign 1, that is from time 01, I don't know if it's clear, from the campaign time 01 till T7, so beginning of the second, we have a different biodiversity from the other two campaigns. A biodiversity that is elevated through the time. And what we can see from the alpha diversity indexes on your right, so Shannon and Simpson, there is a higher biodiversity in the environmental sample in comparison with the one of the food. So, if this is the case, what if we see inside the food matrices, the food composition? And you can see that on the profiles on the bottom right. What we can see, let's take as an example the milk powder. The profiles seem to remain similar in the biodiversity, in the composition of biodiversity, through the time. And more or less this is happening that also to the other material foodstuff. Taking just the time and all the type of sample together, we can see from the alpha diversity indexes that there is an oscillation trend where we observe, I don't know if you can see that we observe in the end of its campaign and elevation in the biodiversity. So here, here, and here. And the same for all the indexes, more or less. But who are the most abundant taxa? If we divide our cases always on time, because this is significant, with red you see the foodstuff, and with blue color you see the environmental samples. For the cases of interest that are Bacillus, we can arrive just in the genus level, Bacillus staphylococcus, and Clostridium perfringens, you can see that we can find them in both, at the both time period, in both type of samples, or just in environment or in food. For example, if we go at the Bacillus, we have it at the first experiment. Then it's not there anymore. Then in the third time we have it, and then it seems that it's entering, and this is interesting, it's entering in the environment, it is remaining in the environment. And so it seems that it's a cycle of entering and going out. So that is exchange, as expected, is an exchange of food and environment. As for example, the Ralstonia piqueti, that is an environmental microorganism, you can see that it is present in the environment and is coming from water origin, and it's remaining plastic pipes, for example, this microorganism. So it's entering from the environment, and then we can see that is also in our foodstuff. So maybe this is something relevant. But, okay, this was the most abundant taxa, but here we have the shared taxa in between the different types of sample. On your right, you can see all the samples, all the taxa derive from all different type of sample, and we can see that the shared taxa are 47, inside of which we have 3 of our interest, Staphylococcus aureus, Bacillus, and Clostridium perfringens. We can see a difference in the shared taxa between the campaign 1 and the campaign 2, that in campaign 1 we have more, but they are always the one that we are interested in. They are inside, they are included the one that we are interested in. But, okay, we have seen all the untargeted methods. What about the results of the targeted method? So here you can see 3 different representations of the results for the outcome of the real-time PCR for the 5 pathogens, foodborne pathogens, that we consider them alive because we have detected them after the 24 hours of enrichment, but also by making the delta, so the difference in between the cycle of exit in the real-time PCR with the time 0. So all the numbers that you can see here, so the 65 for Bacillus, 63 for Clostridium, etc., they are all alive bacteria detected by real-time PCR. So the representation is that in the loop, you have the percentage of them on the left of the environment, on the right of the foodstuff. Here, on the table, you have absolute numbers that they are following the order of raw material, intermediate, final, and environmental. And in the graphical representation, you can see the absolute numbers graphically. What we see, obviously, is that Bacillus, so the two spore-forming, Bacillus cereus and Clostridium perfringens are the most present, followed by Staphylococcus aureus, all of them present also in foodstuff, not just in the environment, and not just in the raw material, but also in final product. And the ones that they are not, and also not expected, is the Listeria and Salmonella species, that they are present in the environment and in some of the raw material, so in fewer numbers. But what if we want to correlate the targeted and untargeted methods? So, to put together the taxa, the abundances that we have for the metataxonomic analysis, and the results, the positive results that we have from the real-time PCR. If we focus in the alpha diversity indexes, and more specific in the phylogenetic diversity, we can see a clear significance, a really strong significance, positive significance, between the phylogenetic diversity and Bacillus cereus. In few words, what I want to say with that if we have a sample with a really high biodiversity, we expect to find Bacillus cereus. It's possible to be that correlation. Going to a linear correlation, so the heat map that we have on our left, says something that is, it wasn't expected, but is the truth. For the cases that we want, that we have detected Bacillus cereus, so on the horizontal line, you can see the result of the pathogen presence from real-time PCR. And on the epsilon axis, you can see the taxa, the abundances. So you can see in the green boxes that when we are going to correlate the two results, we don't have a positive, not a correlation at all for the pathogens that we're searching for. So we cannot use that method at this point. You can also see the same representation in the bar plots where, for example, in the Clostridium, as you can see at the time 8, T12, for the metataxonomic analysis, we don't have any result for the presence of them, but we have a really important presence in all the type of samples from the real-time PCR. We try to find correlations by changing the type of correlation. So not a linear, but a sparse one. This type of analysis, this algorithm, gave us 6 groups of correlation. The one that is interesting for us is the blue one, the third, that contains, if we zoom on it, contains 3 of our pathogens of interest, that is Bacillus, Clostridium perfringens, and Staphylococcus genus, that practically is confirming what we have already seen with real-time PCR. If one is present, we are expecting also the other 2 to be present. It means that. So to conclude, the microbial ecology is changing a long time for all type of samples. Raw materials seem to distinguish from each type. The targeted pathogens were detected with both methods, metataxonomic analysis and real-time PCR, although it seems at this point that their direct association is not an easy issue. Real-time PCR gave us the possibility to identify the presence of alive bacteria. Bacillus cereus, Clostridium perfringens, and Staphylococcus aureus were the most present pathogen. And about the future perspective, we think that with more advanced mathematical approaches, maybe we can define better possible associations. We have still to work on the massive number of isolation that we have collected from the after 24 hours of enrichment by following whole genome sequencing characterization. And also we think that with the metagenomic analysis may have a lead in between microbiota composition and pathogen presence. This work is part of the European project SAFI that is funded from Horizon 2020. I would like to thank my professor and my colleagues for their help until this point, and of course the company for the collaboration. Thank you.
Thank you for your presentation. We're running out of time slightly, so we can afford like one quick question. No questions? Okay, thank you.
Thank you for attending the session.